today I will be presenting how to utilize these templates for an effective workflow. So we went live with the we call it a node, but with the ELN, with Signals ELN, back in May, and so far we've been utilizing it for creating these workflows that are really helping us with our work. So here, I just want to quickly present basically the team and the workflow that we have. So I am in the team of it's called the cytogenomics at the Astellas Institute of Regenerative Medicine. And in the workflows that we are generating, we're mainly using them for RNA sequencing experiments. So a typical RNA sequence experiment, it just comprises of the extraction of the reads of the RNA, and then the library preparation, sequencing, mapping, and then at the end of the run is the analysis part. So the workflow really requires a lot of attention. I mean, right now we are 8 people in our department, and what we're trying to do, so the tasks are really assigned to, we say, okay, so somebody is going to be doing the library prep part of the experiment, and maybe another team member is going to be assigned the sequencing part. Another team member will be assigned the mapping and the transferring of the sequencing, at this point the reads, into our cluster, and then another person will be assigned analysis. And we were struggling how to keep everything in track, so we wouldn't miss any samples, so that we can really look at the entire life cycle of that, just one particular experiment. So yeah, like I said, like multiple individuals may be involved into this multitask. So just to ensure that the experiment was performed just to the entire life cycle, we wanted to create this audit trail for each sample, and that's where we discovered that with this the workflow on the ELN on Signals really helped us with this effort. And so Jamie and me, we created these templates, and the templates really allowed us to generate this experiment that has all the things, well, most of the things that we need. We're still optimizing this template, but so far it has most of the items that we need to really look at the experiment from the time that we acquire the sample to the time that we finish this iterative process of the data analysis. Okay, so so the templates are used to ensure this consistency of the operational workflow, and so each template should include all the essential components. So that's what we were thinking. We wanted to make sure that the template, once we are creating an experiment, we're utilizing a template that will have everything we need for this experiment, or at least most of the items that we need for these experiments. Okay, so we wanted to record the communication between the requester and the individuals that are performing the assay. And this is of particular importance because for us, when we are running and then analyzing sequence run, we are a team, but the requesters might be from different teams. It might be even from teams that are outside of our institution. We have requesters from California. We have requesters from Japan. And the beauty of A-Node is that we can communicate all the different ideas, and then we can put them in this experiment. So all the ideas, all the discussions will be part of the experiment. So at the end, whenever we close this experiment, it will have like the entire package. And that's what we wanted to have or to create. Okay, so yeah, so it needed to be accessible to all parties involved in the experiment. And like I said, we have multiple institutes and from all over the world. And also, we wanted the ability to be edited, especially in the analysis section, since this analysis section for RNA-seq experiments will really go into multiple iterations prior to being finalized. We can say, okay, so this is what we think the analysis is are your targets that we found, number of count. It could be something as small as, okay, we wanted to know what is the differential expression of, sample A versus sample A versus sample B. But then the requester might ask another question, like, for example, okay, so what about, I see that you gave me a target that is differentially expressed between these two samples. But what about, can you add a third sample and then compare those samples? And then with a note or ELN, we can do that. So we will put that information into the experiment, and especially we can say, okay, so this other sample came from this other experiment, but we're also adding that to the analysis. And that really helps. So then we can we can click back to that link, and then we can know what experiment that sample came from. Okay, but in the experiment also we'll capture all the steps that are performed and name the individuals that are performing each task. And again, this is very important because we wanted to know that all tasks were completed. Sometimes, or prior to us using the AELN, we were in instances that, the requester would send us an email, okay, so what happened with, where are my samples? And then we would have to try to find out, through the paperwork that we used before, where were we in what step of that, of the life cycle of the experiment. But now with the ELN, we can all have access. Even the requester will have access. It will know exactly where we are in that experiment, and that way that we don't have to be sending emails at all times. And then, yeah, ultimately, we want the experiment to contain the entire life cycle of this experiment, and it should be reproducible if needed. And the template that we created, it's also useful because it can be shared with other members of the team, and it can also be used for other experiments. So, for example, if we generated a template, or initial template was for a single-cell RNA-seq, and then we use that template and we edit it for RNA-seq, so just bulk RNA-seq experiments. Also for other testings that we have now doing in our group. So it's a very useful feature, or at least we found it to be very useful. Okay, so I'm going to be taking you guys through the so basically the section. So we envisioned, when we were discussing this in the initial stages of creating this template, we envisioned at least 3 main sections. Well, initially, we have more than that. We came out that 3 for us would be perfect. So one section is the requester. In that section, we're going to add all the data that is essential for the requester. So the person that came to us and told us, okay, so we want this sample to be sequenced. So they will be able to find all their data in this section. So then we have another section, section number 2, that is a section for the analyst. So the analyst, in that case, as our team and our team members, will be able to go to this section and find out everything that they need to run this sample, or this specific assay will be part of that section. And we wanted to dump proof everything, so then anyone should be able to follow the steps to perform this task. Okay, and in the other section would be all the protocols that are essential to run this assay. Okay, so let me take you. And that's why we created a page in a tab for each section. So let me take you through this. So in the initial section, we have the requester section, and this contains all the important information required from the requester, right? So what type of analysis? Because this may vary. Again, it could be single cell RNA-seq, it can be bulk RNA-seq. Even within this sequencing, it could be, okay, I just want differential analysis, or I want a specific, please tell me the count of the specific gene that I'm looking for. Identification of markers, all that we want it to be captured at least in the requester section. So the requester will be able to put all their needs and wants into this section, and then us, the analysts, will be able to view that and then use that information for the data analysis in particular. Because the library prep and all the other steps should be the same, but it's mainly for the data analysis part of this experiment. Okay, so that part will be in the rationale. So the rationale is very critical for this experiment. That's where the requester will have everything that they need, all these wishes and wants of what they want. They also, we wanted to capture all the metadata. And for sequencing experiments, I don't know if you guys are familiar with this, but for sequencing, the metadata is crucial because we wanted to know exactly where did this, like for cells or samples, where do they came from? How were they processed? Like any metadata that will help us understand why is there a difference or potential differences in each sample. Okay, so the third point, we wanted to have like the instructions. We want to give the requester all the instructions that are required for a good sample, how to prepare the sample, especially this is for single cell RNA-seq. This is very important. How to generate a clean sample that then we can use. And this is important because, the quality of the reads will depend on the quality of the sample. I know it's that the good old adage, garbage in, garbage out, right? So if they send us like a good sample, we'll be able to give them the best analysis possible. And we also have like the Excel sheets that give us this additional information for the requesters to annotate or have like give us a copy of what the sample, what was the particular sample that they were submitting, the date and stamps and all the signatures required. We also have the analysis PowerPoint. So in this section, after the analyst that is performing the analysis of the sequencing, they will start populating this PowerPoint. So the PowerPoint, this is where it's going to be an iterative process. So the initial, okay, this is what I think was a differential expression genes, or these are the targets that we have identified. So that will be populated in the analysis in this PowerPoint. So the requester will review that PowerPoint, and they might have different questions. Okay, so what about this gene, or what about this? Can you give me a pathway that you believe is important in this set of genes or in this sequencing data? So at that point, that will go back to the rationale. So they will add that information on the new discussion in the rationale. So then the analyst will be able to go to that section, review the new instructions, and then go back to the analysis and then reanalyze the sample and then put that analysis in the PowerPoint. So I think this is great. It's great for the life cycle of the experiment, at least this section. And the communication, it really has enhanced our workflow by just having this section alone. Okay. So then we have like all the sample submission prep guidelines, and this is what I was saying. So all the instructions are going to be contained in the experiment. So this allows the requesters to fill out the forms, we have a timestamp, and then any errors can be caught early, and then they can be fixed. Before, we would have paper documentation that if there was an error, we have to like run and try to find whoever was the requester, and then try to get them to fix the form. Or if there was somebody from, like, for example, from Japan, that might take a little bit longer, but now we don't have to worry about this because everything is online. Okay, so and this is something that we have. This is the section number 2 that is for the analyst, right? It's kind of work in progress. We do want to like fully automate this, and for that we're trying to figure out how to create this, use the tasks to create assignments within the system. And ultimately, we would like to create a system that allows us to effectively communicate and update the status of the experiments to all parties. And what we envision is that as soon as we receive the sample, we would create this task that will send an email, because I know it's possible to the ELN, to the signal, to submit an email notification to whoever are the analysts that were assigned for each task. And then once they are once they complete their task, they should be able to press the completed task tab, and then we should be able to receive an email that task was completed, and then we can go to the steps, to the next steps. But anyways, this is just work in progress, but that's what we're working on right now. And so in the section number 2, the analyst section, so we have this all the documentation, like I said, to make it easier for the analyst to perform all the tasks. So this I'm showing an example of the or workflow that we use, and then we can use like a tablet, for example, to update all the experiment and all the tasks performed. So we go into the form assay, and then we can update the lot numbers that we're using for all the reagents. We can put the sample names, catalog numbers, and everything that we are doing for each experiment will be annotated in this document, and it can be updated live. And also the requester can also see this or annotations or changes live as well. And so a work in progress. We're also trying to use the materials table that may allow us to better capture all the metadata associated with each reagent. But this is also work in progress so far. Okay, and section number 3, we have all the protocols. So all the protocols that are essential to perform this experiment are placed in this section. So as we are running, or any analyst is running each of the steps, they'll be able to access those protocols and then open them in the tablet and actually follow them as they are performing the experiment. And as they are performing the experiment, they can go back to the previous step and input for example, we need timestamps or when each step was performed, and check log numbers, like I said, and also give a time when the experiment was completed. And so we also have this like links that are useful. So for the analyst, especially when the library prep step, that we have included here in another section that we just call useful links. But so in conclusion, I think the Signals ELN really provides an end-to-end solution for the entire life cycle of the experiment. It allows for live quality control for sample submission data. It allows for comprehensive documentation for the experiment execution, and it has this continuous analysis capabilities that are very user-friendly for us and the requesters as well. And it really is boosting the efficacy, right, and reduces the risk of accidents that we have previously had issues with. So yeah, so far we are very content and pleased with our ELN workflow, and we are improving it. Trying to improve it as much as as much as we can, and yeah, that's what I have for now. Thank you for your attention, and I'm just going to acknowledge the leadership over here at the Astellas Institute of Regenerative Medicine, Hida and Joe, as well as all the teams of the cytogenomics, Marco, Jamie. Jamie has helped me a lot with this workflow. Tracy, Abby, Mike, Melissa, and Craig. Thank you so much for